Hey everybody. So you might have heard of a disease called sickle cell anemia before. Sickle cell anemia. It's common in Africa and a few other countries. Well, a few other regions of the world. What happens in this condition is that people who are affected, their red blood cells turn into this curved shape like a sickle, which is a tool that they used to use to cut grass and crops. And it affects the way the amount of oxygen you can carry in your blood, and there's a lot of other side effects as well. And the reason for this is because there's a mutation in the hemoglobin gene in people with sickle cell anemia. And it's actually in the beta hemoglobin gene. There's actually 2 hemoglobin genes, but we don't need to worry about that now. So there's a mutation in the beta hemoglobin gene, and it causes a mutated hemoglobin protein. As we've learnt before, that a mutation in the genetic code will change the protein, and it might cause the protein to lose its function completely, or it might cause it to have a reduced function like it does in this disease. So here, the mutation in the normal genetic code is at this exact point here in the gene. So where there should be a GAG, so this is the DNA strand or the coding strand, sorry, we've got year 7s going past at the moment. So there should be a GAG on the coding strand, and that means on the same thing on the mRNA, it should be GAG. And then on the hemoglobin protein, this is the correct amino acid that should be placed here. And GLU stands for glutamic acid. It's just one of the 20 amino acids that we use, that living things use to build their proteins. Okay, but the mutation, it changes just one letter in the genetic code. So from GAG, it goes to GTG. So there's the mutation. So on the coding strand, it's a GTG, and then on the mRNA, it's a GUG, because remember, mRNA uses uracil instead of thymine. And that one letter change is enough to change the amino acid in that position from glutamic acid to valine. Just another amino acid. But then that different amino acid means the protein changes its structure, and they actually all clump together, and it causes the red blood cell to have that sickle shape. Alright, so that kind of mutation where just one base in the code changes, that's called a small scale mutation. And they lead to single gene disorders, which we learnt about earlier on in the last topic. That's the end of the day. So that's what we're focusing on in this video. But remember, there are another type of mutation, which are called large-scale mutations. And they're the ones that involve whole chromosomes changing or having too many copies of a chromosome. And remember, they're the ones that cause chromosome disorders. So we're focusing on small-scale mutations here. And there's 2 types that can happen. You can either have just a single change in the genetic code. So a single base change. And these are called point mutations. or you can have multiple bases in the chain in the code change as well. Single base changes is what we're focusing on, so point mutations. And there's 3 types of point mutations that can happen in a gene. So let's say this is our gene here. The top strand we'll say is the coding strand. So that's the actual code that will be on the mRNA as well. Well, the first mutation that can happen is just if one letter gets substituted for another, just like in our sickle cell anemia example. So this type of mutation is called a substitution, where one base in the code is substituted for another. All right, these can change the amino acid, like we saw with sickle cell anemia. Sometimes they don't, so they can have no effect at all, leading all the way up to they can have a large effect on the actual protein at the end. So depending where the letter is and how it affects the amino acid, that's going to determine how much it affects the actual function of the protein. So let's say here in our coding strand, we had the T, thymine. It might be swapped out for a G. And then that would also mean that on its complementary strand, that A would swap out for a C, cytosine. So that's substitution. Another type of point mutation is if a single base gets deleted. So let's say let's say a different one. If one of these C's, cytosines, gets deleted, then that pushes all these other bases one slot to the left. So that kind of mutation is called a deletion, surprisingly. Oops. One base is deleted, and all other bases beyond in the sequence move one space up or further to the start of the sequence. So deletions are pretty simple. The other type is opposite to a deletion. You can have one base can be inserted in the sequence by a mutation. So let's say in between these two C's, we might have an A, an adenine. Well, then that's going to push every base that's beyond that one spot further away from the start. So that's going to change the whole code after that mutation as well. So those bases are called insertions. Sorry, those mutations called insertions. So a base is inserted into the sequence. So substitutions can have no effect, or they can have a large effect. Deletions and insertions always have a large effect because they don't just affect the position where the base changed, they affect every single base beyond that in the sequence. So nearly always have a large effect on the protein. Usually they cause the protein to completely lose its function at all. Usually causes loss of protein function. So insertions and deletions are a really common way that genetic diseases can happen, because if an allele has an insertion in it, that allele is not going to be functional, so then if two parents with that allele pass it on to their child, it's probably going to have a recessive condition related to that protein. Okay, that's 3 types of point mutations.